Tommy, and thanks to ARM for inviting us to speak today, and good to see everybody here in Barcelona. Thanks for taking some time. I'll talk a little bit today about Forge Biologics and our CDMO hybrid company out in Columbus, Ohio, back in the United States, and give a little background on what we offer and how we built the company over the last 2 years. So here's a snapshot of our facility in Columbus, Ohio. We've grown since 2020 to 320 employees. The majority of those are in our technical, clinical, and quality aspects of our business. And as you see over on the left, we are a hybrid company. We are primarily an AAV CDMO. We're producing high-quality vector for quite a number of clients. I'll review our overview of our client base in a bit. But as you can see on there, we're also a therapeutics development company. And today I'll be focusing more on the CDMO, but just to give you a little bit of background, we do have a two candidate pipeline on our therapeutic side. Our lead candidate, FBX 101, is a AAV for the treatment of Krabbe disease, a very high impact and high mortality disorder. And this allows us to continue being drug developers, but in conjunction with that, we also can use the tools that we offer to our clients to actually bring them through regulatory bodies first, FDA, EMA, and really get a read before clients actually have their hands on the tools. And I think that gives them a bit of relief around, really what are the sort of the run rate of these tools and what have been seen and what is the feedback you're seeing from regulatory authorities. So we think that business model is actually quite advantageous, not only for patients, but also for our clients as well. And where this really comes down to is culture and team. And CDMO, we find that we spend a lot of our time building culture internally. And the goal of that is to make sure that the people that we have, high talented, high demand folks are retained and make sure that clients have the best experience they have. They see consistent faces. They have the same team as they're shuttled through their program. And it really comes down to the leadership team we built. And we have over 200 years of gene therapy leadership between myself and Tim Miller, our CEO, David Dismuke, our CTO, who's had a long history developing AAV therapies back to Jules Maslowski's lab and all the way through groups like Stride and Voyager and Bamboo. Maria Escalar is running our therapeutics division, and Chris Schilling below, who ran Nationwide's regulatory, is now heading up at Forge. And he's done dozens of INDs on the AAV side. Why we carry a head of regulatory and a regulatory division, we also can help clients with their regulatory, everywhere from advice to publishing to filing. So it really gives them an option to try to keep as much of their work in-house and in one group. So before I talk a little bit about our offerings, I'll talk first a little bit about the field, and I'm sure we've all been paying attention to sort of how the growth of cell and gene therapy has exploded over the last few years. Even with economic downturns, we still see strong pushes for new candidates. Tim Hunt from ARM presented some data I'll share shortly about how the sector has been growing. We have performed our own independent third-party evaluation of what does AAV specifically look like. Obviously it's the most widely used viral vector on the gene therapy side for delivery of genes of interest, but really how is the growth sort of moving along here? And just looking at the growth of clinical trials starting in 2015 and projecting out to 2026, we still see a strong demand and actually a doubling of the number of clinical trials that will be executed. So starts coming into the thoughts about where is all this development occurring? Obviously there's plenty of groups who are doing this in-house, but how can CDMOs be contributing to this and helping with the overall manufacturing demand? And it brings us to a question about how do we address all that manufacturing demand. There's obviously quite a few options, from a manufacturing standpoint, in-house, multiple CDMOs, partnerships. And so we take a step back, we think about how are we going to address the need. And for example, the example that you see here, let's just look at one rare disease, for in this case, DMD. So if we look at just quarter of the prevalence in just the US alone at 10,000 patients, and the amount of titer we're gonna need to treat just a quarter of them, you're looking at roughly 750,000 liters of AAV that will be needed just to treat that one quarter of that one disease just in one region of our planet. And so that equates to a little under half of the available capacity that's in the US alone. So that starts to ask a question about that's one disease, and then Tim presented early in the year. Here's sickle cell, hemophilia, SMA, beta thalassemia. We do have what we consider large rare disease populations. So really we start thinking about what's the best way to address this, and really the idea is how do we scale out our manufacturing capabilities worldwide. So that was the question we asked when we started Forge, is how can we contribute to that and have a developer's mindset come in and then help other clients move their way through and address these large needs of capacity and move their programs through both process development, analytical development in a quick way. And so we've created Forge and our facility here called the Hearth, and I'll show a map of this momentarily. These are some pictures of our technical groups at work around. You see a build on the left. This is our newly produced 16-suite GMP space that we've just completed. Is actually now going through final environmental qualification, and this will be available this year for clients to use. Really, again, with the idea of expansion in mind. So let me tell you a little bit about our current client population. We're now, after a short 2 years, serving over 35 clients, and they really are broadly distributed between sort of private biotech, those emerging Series A coming out of stealth mode, but also quite a number of public biotech, large pharma partners are now sort of looking at entering gene therapy in a large way, looking for a core to do so. And we really enjoy our work with advocacy and academic groups as well, and that's something we bring in and help move those programs along and give advice from a regulatory standpoint. Our platform, we are producing this under an AAV platform developed internally by David Dismuke and his teams. This is all the way from development through upstream, downstream, and filling. And I'll talk a little bit about the tools we use in our platform, but right now, out of the over 35 clients we're currently servicing, over 90% of them are already using our cell line. So, it gave us a lot of great validation that the cell line we produce has got excellent traceability from a regulatory standpoint and has high performance when it comes to manufacturing and yield of AAV. We have over 80% using our PEMBER ad helper, so also using our plasmid options, and 50% of them have started with we have a core offering basically called Blaze, which is a small scale, off the shelf, try it before you buy it, approach where you come in, you work with Forge, you do small scale work, and really look at some of the early results of your constructs. Look at different promoters, look at different GOI assemblies, and come through and collect some results and see where you're at in a relatively quick turnaround, just a few months. 50% of our clients started there and then upgraded to a GMP. So not only is it a wonderful tool for developers to start making early decisions around their program, for us, it's also a great way to introduce them to the team and see the capability to scale up to internally using the same process to GMP. Over on the right, we always get a lot of questions, what serotypes will you work with? And the answer is all of them. We've had wonderful AAV scientists throughout our organizations who have internally at Forge worked on all the wild types and also some variants also on novels, and we're seeing more and more of that now, novel serotypes coming through, looking for robustness and how to produce them in a scalable way, which we can also offer help from the process development standpoint and help them do design experiments to run them through and see what's the right buffer concentrations, what are the right plasmid ratios, these sorts of things are things we can do internally in Forge. So what do we offer? I think I've touched on a lot of them, but one of the beauties of working with us is our facility in Ohio is centralized. That's where all of our operations are located, and we can produce all of our offerings under one roof, and we can do them internally as well. So everywhere, starting with our research. I mentioned Blaze. This is our off-the-shelf research offering. This is a great way from 1 to 40 liter scale to start collecting some early data. We actually last year also now offer plasma manufacturing and have numbers of clients who've jumped on this relatively quickly for supply. We only produce plasmids for our clients. So it is a boutique benefit of working with Forge to then have your own dedicated queue to get your plasmids produced. We're doing them at 3 grades, so similar to all of our experiences, research, a mid-grade for phase 1 and 2, and then we have a GMP grade that we'll be bringing online later this year. Also, full process and analytical development capabilities up to 500 liters. We have all of our assay development is in-house. Over 20 assays here are produced at Forge. So we don't sub except for some specific microbiological assays that we just don't want to bring in-house, or a commodity item that we can use at one of our partners, but we can manage that for of our clients. And then that also includes GMP manufacturing release testing as well. And then we do have 20 GMP suites we're using for GMP manufacturing, ranging from 50 liters up to 5,000. I'll talk about that shortly. And all single-use closed systems. And also we offer filling capability as well, including automated. So the goal here was really try to create a situation where you can come in, a lot of small companies who are still working through their ideas. Once you've sussed out sort of what exactly is your candidate going to be, help you produce toxicology lots through your preclinical work, and then the goal then to GMP. So you've had the same cells, plasmids, materials construction, everything all the way from development straight through to your IND approval and beyond. So how are we going to do this? Sort of the master plan of Forge here, and I think I've touched on most of these. It's really mostly about scale out and scale up. Our goal here is to be able to service AAV customers to have plenty of capacity to produce their future products, thinking about commercial and what those needs are going to be based on earlier slides, and then thinking about cost of goods, thinking about reimbursement. How are we going to drive down cost price so we can increase our volumes, but also service a larger rare disease population? So that's the goal of our scale-out plans as well. And obviously, we continuously improve the process. We have our own internal molecular development team. We actually can do base pair analysis from sequencing. We can go back and look at GOI structures and why things may not be constructed correctly and help clients as early as that discovery step. And obviously, enhance analytics. As we're starting to consider move towards our BLA customers, CQAs, CPPs, looking at critical process parameters, building those out, having those part of our platform, trying to make Mod 3 as easy as possible in the US, and also EMA filings from a CMC standpoint, and try to make them as consistent as can be. So quickly on the facility, here's a map of what we have. As you can see over on the left, we have cGMP manufacturing suites in the upper left. There's 4 there, 2 upstream, 1 downstream, and a cell culture suite. We built that very rapidly when we started the company in order to really get to GMP manufacturing, intended for phase 1, 2. The space in the middle, you see the GMP manufacturing is our newly built and now operational suites. 16 of those, and they are designed actually for late stage and commercial. So we have background cascades, background classifications that meet both here in the US and around the world in the EU. We have also quality control space that'll be dedicated to that as well. You can also see process development space on the right, R&D, analytical development, and plasma manufacturing. So we have that all right in-house, and all under our CTO, creating all the development work, and of course utility support, and our own GMP warehouse. So we do carry our own supplies. We don't order upon demand. So if a client comes in and says, I want to start, they don't need to wait 4 to 6 months before all of the materials come in. We have that on site. And in referring to scale out, so we can go all the way up to 5,000 liters. We have developed a 5,000 liter process with the thermodynodrive, and are moving that through clients now and continuing the development with those. And we can actually scale down as well. So microcell technology using Amber 250 and down to 1 to 10 liter scale. As we're starting to get requests more about how do we actually characterize aspects of the process we'll need for our BLA, we want to make sure we have a consistent and robust scale down process as well. And then our process improvements and how is our scales looking. And just one example of some of the data we've collected about yield going from 500 milliliters to a liter up to 1,000 liters. And we continue to see consistent yield. Always one of the concerns with scale up is loss of titer and yield as you continue to increase. But luckily, we have seen over multiple serotypes a consistent yield, which is something that gives some confidence about when we scale out. And we're trying to service populations that we can continue to do that on a scale that looked very similar to PD as well. And just briefly in the last minute or so, our platform assets, we have our ignition cell lines. This is our HEK clonally derived cell line, developed in-house at Ford, so we don't license this. We do have a license back to the original HEK cell line, as most do, but this was developed in-house by David Dismuke. And what we think is unique here is we tried a different approach on this. This is a royalty-free cell line. You work with Forge. You produce a GMP batch with us, you can take the cell line if you feel like you need to take it in-house or elsewhere. Obviously, we hope you continue to work with us, but it keeps away a lot of the reins that you may have on another cell line of where you can work and how to move it around and what sort of payment structure's there. This has one of the best well-documented history I've ever seen on a cell line. No big black boxes, so when you go to the agencies, we have the data back to origin to show where that cell line has been and how it's transferred. So that's helpful from a capacity standpoint and from a regulatory standpoint. And then to our PEMBER, we've created a ad helper that is significantly smaller than those that are available on the market. So we did this to remove certain elements from the ad helper that could be considered sort of potentially a regulatory target such as fiber units and such that we think that would be best to have out of it and actually functionally still works the same. Our data have shown that in comparison to other to another industry available ad helper, similar purity results. And then when we go through our full and empty ratios after elution and even as early then around 43%, but after full concentration at around 93% heading into drug products. So really high purity, empty full ratios obviously being a big topic as we move through the AV history and something we watch very closely through our own analytics. And then finally, just a list of analytics we offer, all in-house, except for a few like sterility, mycoplasma. These are things and adventition agents we don't offer currently, and likely we'll keep out some of those purity assays and impurities you see at the bottom. They're coming in next year into the facility or potentially even later this year. So then finally, last slide, how are we working at ex-US? We now have clients around the world, in Europe and in Asia as well. So we have a sub here in Spain. So we actually have the ability to work locally with clients here in the EU. We have our stage 2 facility has been designed to meet EMA background standards, thinking about conducting clinical trials, and also commercially having product here in the EU, working on our cold chain routes for distribution because of those clients. And QP is something that we've wrapped up too in the facility, and we'll be porting more on, even having that expanded to our new facility. So that'll be also for clients' use as well. So thank you all very much for your time. If you're interested in hearing more, we have a booth outside, and if you see myself or any of my colleagues here, we're happy to share a little more on the Forge story. Thanks for your time today.